So, transcription is the first step in decoding the genetic information of a cell. During transcription, enzymes called DNA-RNA polymerase build the RNA molecules that are complementary to a portion of one strand of the DNA double helix. So, the DNA helix will have been opened up in a small section called the transcription bubble. Now, RNA molecules differ from DNA in several ways. A, they're single-stranded rather than double-stranded. B, their sugars are ribose, not a deoxyribose. And C, they include uracil nucleotides rather than thymine. And because they are single-stranded, RNA molecules don't form helices. So rather they can fold up and form complex structures by internal complementary base pairing, and we'll see an example of that with transfer RNAs, which are involved in a later stage of protein synthesis. So the three general classes of RNA molecules which are involved in expressing the genes are messenger RNA, and they carry the coding sequence for protein synthesis. We call these transcripts. And the ribosomal RNA molecules from the core of a cell's ribosomes, they're also made of RNA, but they're found within a complex of proteins, and they form the structure of the ribosome. Then we have transfer RNAs, tRNA, and these molecules carry amino acids to the ribosomes during protein synthesis. So in each class of RNA, it has its own type of polymerase. This is only true in eukaryotes, and we don't see that in prokaryotes. We also know there are other types of RNA, but we don't fully understand what their role is within the cell. Maybe to do with regulatory roles, maybe to do with protection against viruses. We're not really sure, but we do know that they play a part. So back to our messenger RNA. It's the most variable class of RNA, and there are literally thousands of messenger RNA molecules present in a cell at any given time. Some molecules are really abundant, and you might find them in hundreds or thousands, and that's often true of the transcripts of structural proteins. However, other messenger RNAs are quite rare, and you might only find a single copy present, and that's more likely to be things like signalling proteins. So messenger RNAs also vary in how long-lived they are, and in eukaryotes, transcripts for structural proteins might remain intact for like 10 hours, whereas transcripts for signalling proteins might be degraded in less than 10 minutes. So cells can be characterized by the spectrum of the RNA molecules within, and this spectrum is called the transcriptome. Whereas each cell in a multicellular organism carries the same DNA, the genome, its transcriptome varies widely according to the cell type and function. So, for instance, the insulin-producing cells of the pancreas contain transcripts for insulin, but bones don't. So even though bone cells carry the gene for insulin, it's not transcribed. Therefore, the transcriptome functions as a kind of catalogue of all the genes that are being expressed in a cell at a particular point in time. And this can be really useful in research when we're trying to identify particular functions of an unknown protein, and we may find it expressed very highly in one region of an organism and not in another. So that's the transcriptome. And transcription is the first main stage in protein synthesis. And for today, I'm going to leave it there. Next, we're going to talk about the function of the ribosomes, and translation, the final stage in protein. Well, it's not really the final stage. It's the main final stage. There's always a little bit more with biology, right? It's never quite the end.